We also have a study that is almost identical design with Ibrutinib, Bendamustine, Rituximab plus Ibrutinib. That also showed a highly significant difference in progression-free survival in favor of adding the Ibrutinib. So it's clear to me that if the thinking is, okay, I'm going to give Bendamustine and Rituximab, yes, it's critically important to add a small molecule inhibitor, and I would either add Idelalisib or Ibrutinib, again, depending on the clinical situation. Now, both studies were missing an arm, and the arm that they were missing was, what about forgetting the Bendamustine, just Ibrutinib and Rituximab, or Idelalisib and Rituximab, and forget the Bendamustine entirely? Is that a competing substitute for Bendamustine Rituximab? And that's a great question, which we didn't ask in either study. And because it wasn't asked in either study, we don't know. If you do that thing that statisticians hate, and that is the cross-trial comparison, you might say, well, gee, the results with single-agent Ibrutinib or Idelalisib and Rituximab look really sort of similar to the results with the Bendamustine, so maybe, but the populations are a little bit different. It's dangerous to do those cross-trial comparisons. I think with the Bendamustine Rituximab studies, we know that Bendamustine Rituximab can be improved upon. Whether it can be replaced, I'm not sure we're ever going to see the formal trial to ask that question, but certainly the big challenge in the field is no longer, what works? We have new interesting drugs, but these drugs are exceedingly expensive and they're given in an open-ended way, and that's just not affordable. You take rich countries like the US and it's barely affordable. But around the world, open-ended treatment with drugs that are costing more than $100,000 a year simply is not going to be possible. But because we have a series of new drugs, and in the US we just actually got approval of yet another drug for CLL, Venetoclax, and between Obinutuzumab and Venetoclax and Idelalisib and Ibrutinib and several other molecules that are coming on, we know we have a number of novel agents, and the key is not to use them one at a time, but to figure out how to best combine them so we can get durable and meaningful remissions. And that's really where the next step is. And certainly I encourage clinicians and patients to participate in these upcoming novel studies that are looking at combinations of these agents in order to achieve the goal of eradicating minimal residual disease and actually stopping treatment.